Hi guys, today we're going to be going over the microscopes topic. This is the second video to our microscopes topic. In the last video we went over the types of microscopes and key definitions. In this video we're going to be going over staining, slide preparation and biological drawings. So first up, what do microscopes actually do? Microscopes allow us to view specimens, which are things being studied, that are too small to view with the naked eye alone. Microscopes magnify images to make the specimen appear larger so that it can be studied. They also have a resolving power. This measures the strength of the microscope's resolution, visualizing specimen. Many specimens being studied will be transparent, and so light will be able to pass straight through the sample. This means it will not be able to be seen. Stains, which are usually dyes, will be used to visualize a sample being studied. Different stains will visualize different subcellular structures depending on what you want to be able to see. For example, methylene blue is a stain that's used to turn DNA and RNA in cells blue. There are three ways you can prepare a sample to be studied that you will need to know about at A level. You can do a dry mount, a wet mount, or a smear. Here's how to prepare a dry mount. In this case, the specimen is placed straight onto the slide. No water or stains are used in this process. The specimen is cut into very thin slices so that some light is able to pass through the sample to allow it to be seen. This is called sectioning. A cover slip is added to the slide above the specimen. Samples such as hair, pollen, feathers, or plant material can be studied with dry mounting. And here's how you do it. First, you section your specimen into thin slices, either using a scalpel or a special machine. This is so adequate amounts of light can pass through the sample. Using tweezers, you then place the sample on a slide. You then place a cover slip on the specimen, and that's it. And now on to preparing wet mounts. Here the specimen is placed directly on the slide. Water and stains are used in this process, as water is used to suspend the sample in. The specimen is not normally cut in this process. Then the cover slip is added to the slide at an angle to prevent air bubbles forming from the water droplet added. Wet mounting is usually carried out for samples of red blood cells, white blood cells, or even parasites. Now we'll take a look at preparing the wet mounts. First, you add a water droplet to the slide using a drop-in pipette. You then add the sample to the water droplet using another drop-in pipette. We're using a blood sample as an example. You then add the cover slip at an angle of about 45 degrees over the top of the water droplet containing the sample. Now a stain is added to the edge of the cover slip. Tissue paper is normally placed at the other side of the cover slip to draw the stain across the slide underneath the cover slip. This will give a thin layer of stain and still allow some light to pass through. Now we're going to look at preparing smears. The specimen is a liquid and it's usually blood. The sample is added to the slide, usually with a drop-in pipette. Another slide is used to spread an even coat of the sample across the original slide. Water is not used, but stains usually are. Smears are usually used for blood samples, semen samples, and microorganism samples. Now we're going to go into carrying out smears. First, you add the sample droplet to the slide using a dropping pipette. You then use another slide and place the edge of that slide on the original slide with the sample on it. You then swipe this slide along and over the sample at a 45-degree angle. This will spread the sample along the slide. You then drag back the slide firmly and swiftly to spread it further and create an even coat. Once you have examined your specimen and collected photographs of the sample underneath the microscope, also known as micrographs, you must be able to draw your sample. This is called a biological drawing, or otherwise known as a scientific drawing. There are rules to these drawings, however, which we will dive into next. And here are the rules you must remember. A title must be included. State the magnification used. Use a pencil to draw. Use white blank paper. Use more than 50% of the space provided. Draw straight lines which do not overlap. No shading. Ensure proportions are correct. Only draw what you can see. Do not guess. What do you think is wrong with this drawing? Pause it and give it a go. Here's what's wrong. Areas are shaded, the label lines overlap, and the label lines have arrowheads. Now that's it for the contents of this video. Now give these practice questions / activities a go. Here's the answers. Now that's it for this video. I hope you found it useful. Please do give it a like and subscribe to our channel. And make sure to look at our channel and see if we post any other videos about any of your other A-level subjects. Bye bye.